Thank you, Anna. Hello, my name is Marina. The theme of this webinar will be investigating gene functions and human disease throughout data on the IMPC portal. So the first part will be about IMPC, our data workflow, and standardized phenotyping pipeline. Then Francisco will show you a new rebuilt website and types of data that can be found there. And then the next speaker will be Diego. He is from Queen Mary University of London, and he will give you introduction to Phenotyping and how new model of human disease gene association can be found. What is the International Mouse Phenotyping Consortium? The IMPC is an international effort by 21 research institutions across the world. The aim is to identify the function of every protein-coding gene in the mouse genome. You can see all the centers in the map here. About our data processing workflow. Mouse clinics generate data across the world. Then it will go through the data coordination center where data validated and go through the quality control checks. We use additional resources such as Gentar and Impress. Gentar is a system of tracking the production and phenotyping of different types of mutant allele that IMPC generates. And Impress we will discuss later in more details. After preliminary data generated, it goes to the core data archive where statistical analysis is running. Then Queen Mary University of London ran human disease pipeline to generate human disease association, and they use Phenotyping algorithm to do this. Diego will tell you more about Phenotyping and human disease associations later. After that, all the data goes to the mousephenotype.org, where data can be freely accessible by everyone. The current data release is data release 22. About IMPRESS. IMPRESS stands for International Mouse Phenotyping Resource of Standardized Screens. IMPRESS contains standardized phenotyping protocols and has two main functions. The first function is that Impress defines pipelines and protocols regarding how they are performed. And the second is Impress provides validated information in both human and machine-readable formats. So all measured and data collected by international mouse clinics as defined by these protocols. Standardized phenotyping pipeline is applied for 3 stages: embryonic, early adult, which is up to 16th week, and late adult, which is after 50th week. So everything starts with embryonic phenotyping pipeline. Homozygous mutant animal can be viable, subviable, or lethal, and based on the viability, different procedures will be applied. For example, for homozygous lethal animals, embryo-LAC-Z and micro-CT procedures will be applied. Viable homozygous and heterozygous lines then will enter early adult pipeline. Early adult and late adult phenotyping pipeline goes after embryonic. There are a lot of mandatory and optional tests. For example, grip strength, ECG, and X-ray are mandatory procedures. They are shown in blue, and optional procedures shown in white. For example, echo and colorimetry. Our aim to learn as much biology as possible, so IMPC perform different procedures. You can navigate our website to know more about IMPRESS procedures parameters. To identify phenotype from data, IMPC generates two groups of mice. The first group is a control, the second is mutant. We usually generate up to 7 female and 7 male mice, and more than 7 female and 7 male for the control group. Then data goes through the statistical pipeline, and some of the phenotypes assigned manually. All significant hits enter annotation pipeline, where MP term from mammalian phenotype ontology assigned. For the statistical pipeline, we use p-value less than 10 to the minus 4. Here you can see an example of MP term for significant phenotypes. It is body length. It can be increased, decreased, or abnormal. You can see ontology term on the right and also ontology identifier. We also use soft windowing technique because IMPC generate complex data from small treatment and large control groups, and it increases the power of the analysis, but introduce variation over time. To avoid this, we use soft windowing, and also soft windowing allows to reduce number of false positives. As you can see in the chart here, control data shown in pink, we see a lot of control data, and mutants experimental data shown in blue. To make phenotype calls stable over time, we apply a window here around the proximate date of the experiment mutants. Well, in conclusion, IMPC is a worldwide collaboration of research institutes. The aim is to identify the function of every protein-coding gene. The IMPC generates new phenotypic data for 3 life stages, embryonic, early adult, and late adult, in a standardized way. All the data from centers undergo validation, quality control, and several pipelines to identify mutant phenotypes. And then MP term is assigned to significant hits, and human disease associations are generated. And finally, all the data are available on the IMPC website. So now Francisco will give you a live tour around website. I'll stop sharing my screen.
Okay, thank you. Sorry, thank you, Marina. So yeah, I'm Francisco. I'm going to give you a tour of the website. So hopefully you can now see my screen. So to give you the tour of the website, I need to pick a gene. I'm going to pick DBN1. You can follow along, or you can use one of your gene of interest. For now, I just typed DBN1 and press enter. So from the search results, you will be able to find some useful information. We have put the mouse synonyms alongside the human symbol and human synonyms. There are some indicators about like the phenotyping stage, whether it has started, some data has been set, or it has finished. You can see it from the results. As well for the ESLs that I produced, you can see whether these type of tissues have been produced. So for now, I'm just going to click on the result, and I'm going to explain you about each section on the gene page. So you can see from what we can see, the first section is the gene summary. So you can view some information on here, the gene symbol, the name. Most importantly, on the left, we have the list of significantly impacted systems. So we have, in the case of DBN1, we have 14. So cardiovascular, embryo, nervous, skeleton. As well, we have the not significantly impacted. So we only have the icons, but if you hover over them, you will be able to see the names of it. So respiratory, limbs, pigmentation, so on. On the right, we have some data collection links. They indicate whether we have data for that specific section or not. So in this case, DBN1 has data for all of them. Additionally, some users are interested in viability and embryo data, so we have added a few ones in here. And finally, we have from the gene summary, you can see the number of allele products available. In this button, we have 5 alleles, and by clicking on this button, you are able to navigate all the way down to the order section. So for now, I'm just going to continue with the phenotype section. This is the section where you will be able to have access to the vast majority of data produced, and is divided in 4 subsections. So they are represented by these tabs. So the first one is the significant phenotypes table, is where you will be able to find all the phenotypes found that have that are below the IMPC threshold, the 1 × 10⁻⁴. So you will be able to see the phenotype name, the system, the allele, zygosity, life stage, significant sexes, and of course the significance value. You probably have saw that for some of these phenotypes we don't have a p-value, and it's because these are the manually marked as significant. So we have marked across the website these kind of cases, so you always know why we are displaying something as it is appearing. And something also very important to take into consideration is that on this table only appears the most significant p-value, because these phenotypes depend on some experimentation. So to demonstrate you that, I have to search for the hyperactivity phenotype. I was able to put it on the search box, and I'm just going to click on the supporting datasets link. And this is the page that you are able to view all the parameters and all the parameters tested for this specific phenotype. So, okay, it's taking a bit to load. So this is the page where it's going to show a data comparison table where you will be able to find a summary of all the parameters. So you will be able to see the parameter name, the phenotypic center, the allele, zygosity, significant sex, life stage, colony, and we have an expanded p-value with the male and female combined, male and female. So probably I'm just going to fall back, demonstrate you the website through my local version of it. So I just going to need to the hyperactivity one and going back to the same results. So okay, so we have all the parameters that were measured for this phenotype. So we have all the names and the expanded p-value columns, because all the tests are, well, the parameters are tested with the male for the male mice, for the female mice, and some tests are not, the sex is not considered. So by default, the page will show the most significant parameter, and it's going to be highlighted in orange. You can see as well the significant p-values are colored in orange, and the ones that doesn't meet the threshold are colored in black. And to see the rest of the details of the other parameters, you need to click on each row. So you can see that this heading has changed, and it's based on the parameter name. So it's going to give you the details of that parameter. So each parameter will have the description, the number of mutant mice, and the mutant and the mouse controls. If you are interested in knowing what other genes are associated to this phenotype, you can do it through this link. It's going to take you to the phenotype page where you can find that information. Also, for the experiment, if you are interested in knowing exactly the steps of how the experiment was performed, you can do it through this link. It's going to take you to the IMPRESS page. We have more data. So each data type will have their own set of visualization. This is the total of the distance travel. So if you hover over this data, you will be able to see these measurements. So in this case, we are able to summarize by grouping the data by sex and zygosity, or if it's wild type. And on the scatter plot, you can see how the data has been generated. So as Marina showed us on her slide, the pink ones are the controls and the blue ones are the mutants. And as well, we have added a dashed line, so you can know, you can have some idea of the slice that has been used in the sub-windowing procedure. In the rest of the page, we have the statistical analysis results, summary statistics, and the attributes of the statistical method used in the experiment, windowing parameters of the sub-windowing procedure, and of course, if you are interested in further using or analyzing this data, you can download it or have access to different ways in this section. So this will be everything for the supporting data page. I'm going to return to the DBM1 page, and that was the significant phenotypes. The next subsection is the all data table. It's mostly has the same data, but it's for all the parameters measured. So you can find allele zygosity. We have a significant column, so you can see which parameters are the significant ones using this column. The next subsection is the graphical analysis chart. So this chart is a visual representation of all the data table. So on the x-axis, that's going to represent the significance value of the parameters. So everything on the right is going to be not significant. Everything on the right is going to be significant. And we have put a dashed line to represent the IMPC threshold, the 1 times to the -4. And on the y-axis, the chart can switch between physiological system or procedures. You can change it through here. And let's say that you are interested in this kind of data, like the behavior neurological system. So with the control on the right, you are able to check to see in more detail and zoom in onto these specific parameters. You can notice that some icons are different. So these are the manually assigned phenotypes. So we have to distinguish them, we have put another icon. And the final subsection is the alleles by phenotype. So this chart is going to appear only for those genes that have more than one allele, sorry. And we hope that it's going to help you to discover phenotype similarities across the alleles of one gene. So for example, I'm going to click on the first column, and it's going to be about the TM1B allele, and it has these 29 phenotypes. And if I click on the last bar, it's going to be the intersection of the TM1B and TM1A alleles, and it has these 5 phenotypes in common. The chart can switch also between showing specific phenotypes or summarizing across physiological systems. So if I click on the same bar, the one that is the intersection of the two alleles, it's going to have cardiovascular, immune, mortality, and these biological systems. So this is it for the phenotype section. The next one is the LAC expression. So this is where you will be able to find all the results from the beta-galactosidase tests that can be used to assess the gene expression on different tissues. So you can see we have the anatomy of the tissue or the tissue name, zygosity, images, and the ratio of the mutant expression in mice and in controls. Some of these tests have images associated. So I'm just going to open one of these as an example. It's going to open an image comparator where you will be able to see images from controls and mutants. So each image will have some information related to it, like, for example, the age of the mice, the sex, the control, whether if it's the zygosity, the allele, and more information. and you can change between seeing all the images that we have available. So you can do comparisons between different images. So yeah, I have finished with the Laxis expression. So as you have seen, some of the tests have associated images, and in this section, in the associated images, you will be able to find all the available images that are related to the DBN1 gene and are categorized by procedure and sorted alphabetically. So at the top, you can see we have some of the adult LAC images that we saw. On the middle, we have the embryo LAC images, gross morphology images. We have some micro CT scans of the embryo in different stages on the middle, and on the X-ray, we have more scans. So I'm just going to open one of these links, and it's going to give me access to the same comparator where you can see control and mutant images across to make comparisons. So going back to the DBN1 page, we have the human disease section, but I'm going to leave for Diego at last, and I'm going to skip over to the histopathology section. So this is the section where all the significant histopathological findings will appear. It's going to have the tissue, the zygosity of the mice. In the case of DBN1, the tests were performed, but there wasn't any significant finding. We can see all the data by clicking this link. So I'm just going to open it. And after it loads, we can see the tissue, zygosity, a mouse identifier, description of the finding, the ontology term that was used, and some scores about the severity and the significance. If you are interested in knowing more, we have the score definitions on the top of the page. And as you can see, we have some data, but it wasn't significant. So this is for the histopathology section. We have the IMPC related publication, and this is a list of curated publications that have used IMPC data or materials to further study the gene. So you can do the same for your gene of interest. You can always download this information. The external links is a new addition. In this section, we are going to put links to other useful resources. For example, we have the GWAS catalog for now, and we have the order mouse and ES cells section. So this is the section where you can find all the tissues materials available, grouped by allele. So we have all the different alleles here. And if you are interested, or if we have put this phenotype column, so if you are interested, let's say, in the TM1 allele, you can check and filter by clicking on that link. You will be able to see only all the data related to the measured data related to that allele. And just going back, I'm going to click on any material. I'm just going to use the ESLs one, and it's going to take me to the TM1 allele page, where all the tissues, all the materials are available, and you can find the full details of it. So the most important one is the link to the stock center that has that material. So through these links, you are able to order them. So as you can see, we have targeting vectors with its information, mice, a little map, and a summary of all the materials available. So I'm just going to return to the human human disease section, so Diego can continue the webinar. Thank you.
Thank you very much, Francisco. Can you see my screen?
Yes.
Great, thank you very much. Now I will jump into the final section of this webinar, which is the disease section of the IMPC website, but I will also guide you through some of the impact of the IMPC as well as contributions. So far, both Francisco and Marina spoke about the phenotyping pipeline and the statistical analysis that happens. But then what happens after that? How do we go from the mouse models to the human? And this is where the human disease pipeline begins. Within this pipeline, we have two main objectives. The first one is to try and find novel Mendelian gene disease associations with the mice data that we've produced, and also to see if the models that have been generated recapitulate human disease phenotypes and perhaps use these models to study disease. How does that look like on the website? Well, I'll scroll here to the website where I'm interested in a specific gene. In this case, that would be DSG2. Once I look for that, you'll see that the human symbol and the mouse symbol are distinguished there. So in case you're looking for an ortholog, there is no confusion now. Second. Super. If we click on it, we can scroll down to where Francisco left us in the human disease section. It's just taking a bit to load. But essentially what we will be seeing here are all of the different diseases in humans that have been associated with this specific gene. So let's go column by column. The first column is the disease. You will see the name of that specific disease. Then we have the similarity of phenotypes. You can see the score by hovering over the bars. You will be seeing something called the phenotype score. This is a percentage score that goes from 0 to 100. That tells you the similarity between the disease and the model for this specific gene. I will go down deeper into how this is calculated and how to interpret it in the later slides. Next, we have the matching phenotypes column, which are the phenotypes from the disease that have been matched to the phenotypes in the mouse model. And finally, we have the source column, which is where we got this disease from. So in this case, that could be OMIM, could be Orphanet, or it could be Decipher. You'll see two different tabs here at the top. The one on the left will show you diseases that have been curated by any of these three entities I've mentioned. And the ones on the right are not direct associations, but are predicted associations by matching the phenotypes. So anything where a specific disease has matched through the phenotypes to a specific model will appear here and could be quite informative if you're searching for novel disease models, for example. The final part of the disease section is if you go to this expand column. So if you're interested in a disease, for example, dilated cardiomyopathy, you can click on the expand section and you will see what's called the Phenogrid. This Phenogrid is a collaboration between the Monarch Initiative and the IMPC, and it allows us to visualize the disease phenotypes on the left and the different mouse models on our database at the top. You will see the name of the mouse model, and you will see the phenotype score beginning at the label. If you want to explore a little bit, then one example you can use is you can look for the color coding to see the similarity between the phenotypes and the mouse models, but also you can see which phenotypes have been matched between the disease and the mouse model. So if we hover over one of the boxes, in this case, we'll see that for this DSG2 model with the allele TMA1, the congestive heart failure disease phenotype has been matched to the mouse cardiomyopathy phenotype. So I do encourage you to go and explore that with any disease of interest, if you like that. Now let's go back to the presentation. I spoke about that Phenotype score. It's calculated with the Phenotype algorithm, and Phenotype stands for phenotype comparisons for disease genes and models. This is a quantitative measure that tells us how well a mouse model recapitulates the clinical features of a specific disease. And the way we calculate this is through the mammalian phenotype ontology to annotate MP terms, and the human phenotype ontology to annotate the disease. So the way we do this is we take a disease, we take the HPO terms that have been annotated to it, and then we align those with the MP terms from the mouse models in our pipeline. And in this way, we're able to produce that percentage similarity score. Let's look at a graphical example. So now we've arrived at the human disease pipeline. We're interested in this specific disease again, dilated cardiomyopathy. Below, you will see the disease phenotypes that have been annotated to that disease. And then on the right, you'll see the mouse model that was matched to it with its corresponding MP annotations. Now, the first step in that pipeline is we look at which phenotypes are similar. So these will be the phenotypes that have not been blurred. And with those phenotypes, we can go and calculate the phenotype score. And in this case, this is 48.77. So with this, we might agree that this is a nice model that has recapitulated the majority of the clinical features of that specific disease. Now, how do we interpret the phenotype score? I will give you 3 different examples with different phenotype scores so you can understand and interpret when you see one of these scores on the website. Here's the first example. So on the left of this vignette, we have a disease. This is maturity onset diabetes of the young, and it's been matched to a model of PDX1. The phenotype score for this association is 96.08. And below the disease, the boxes represent the phenotypes of the disease, and on the right, the boxes represent the phenotypes of the model. The gray boxes are the phenotypes that are matched between the disease and the model, and the white boxes are the phenotypes that do not have any match. So in this specific case, we see that the disease has all of its phenotypes recapitulated within the mouse. When we have this behavior, when the phenotypes are all enclosed in that mouse model, this pushes the score up. Let's look at the different example. This is a different disease and a different model. This time the phenotype score is 59.88. And out of the 3 phenotypes from the disease, one of them was not found in the mouse model. What this tends to do is it pushes the score down. And finally, we have a more pronunciated example, different disease, a different model, where the phenotype score is 37.73, and only 3 of the disease phenotypes were recapitulated within this mouse model. There were numerous other phenotypes from the nervous, skeletal, musculature, digestive, and respiratory systems that were not found in the mouse. So whenever you look at the scores on the website, look at the amount of phenotypes in the disease, look at the amount of phenotypes on the mouse. That might give you an idea as to how to interpret that score critically. Now into the next section, I will give you some examples of the impact of the IMPC and the different contributions that we've made. So up to data release 22, the latest data release, we know that for the roughly 19,000 human protein coding genes, there are around 5,000 that have Mendelian diseases associated with it. And out of those 5,000, almost 60% of those genes have an ortholog that has entered our pipeline. From those roughly 3,000 genes, just over half of them have an IMPC model that recapitulates the disease phenotypes that we observe in patients. Now I'll give you 2 different examples where specific researchers have used some of the IMPC data. The first one is a case of the gene BBS5, and the disease is Bardet-Biedl syndrome 5. This is part of a group of autosomal recessive ciliopathies that affect multiple organ systems. In this specific instance, this disease is characterized by displaying retinal abnormality phenotypes in humans and obesity. And up to when this paper was written, there was no mouse model for this disease. When the IMPC generated this model, it was found that it displayed some of the key phenotypes from the disease, which are retinal dystrophy and obesity. A second example is a case study, and perhaps more recent, a case study with osteogenesis imperfecta. This is a disease characterized by frail bones. In this specific paper, the patients showed neurodevelopmental features and dysmorphic features, dysmorphic features, sorry. Then they went and looked into the IMPC data, and they found that the mouse model for this specific gene had very, very similar phenotypes as the disease, and this contributed to the body of evidence for them to analyze that relationship between the gene and the disease. The final example I'll show you is a paper we published recently where we wanted to quantify the contribution of the IMPC towards Mendelian disease gene discovery. So what we did was, in that last section that Francisco showed you, we looked at the publications that perhaps used IMPC in some way. So we took the publications from 2011 to 2024, and we looked if they used either IMPC data, so the data that you can see on the website, or the directly IMPC mice. And then with those publications being curated, we went and consulted sources that dedicate to curate those gene disease associations, such as OMIM and PanelApp. And we wanted to see if those publications that we curated were present in the body of evidence either of these two entities use to establish such relationships. From the corpus of papers that we found, we categorized them into two different categories. The first one is Mendelian disease gene discovery, and the second one was functional evidence for disease mechanisms. So what we found was as follows. For the Mendelian disease gene discovery, we saw that 35 genes had publications that used IMPC data, 29 used IMPC mice, and 13 used data and mice. For the functional evidence for disease mechanisms, 1 gene used publications from IMPC data, 29 from IMPC mice, and 2 from data and mice. That's it for the disease section. Now, if you want to get help with any of the concepts that we've introduced in this webinar, feel free to go into the help section, which is highlighted on the top right of our website. You will find a lot of information about further expanding on what we've spoken about today, but also frequently asked questions, how to access all of the data programmatically as well as non-programmatically. If you don't find the answers there, please do contact us. I've highlighted the button there on the top right. You will have to fill out a form and we will reply to you as soon as possible. To complement this webinar, we have launched 2 different tutorials. The first one is a website tutorial created by Francisco, which will expand on some of the concepts that he has touched upon today. And the second one is created by Marina, who has designed this tutorial to help you access the data programmatically from our website. Finally, in terms of acknowledgements, these are the funding sources for our work. And I would like to highlight that last code in bold, which is a notice of special interest award that enabled us to improve the website. I think that's it for me. Back to you, Anna.
Great, thank you very much, Diego, and thank you to all 3 of our speakers this afternoon. I just wanted to highlight that we have the Q&A box that's open if you would like to add a question there, and we will come to the questions in just a moment. Before we do, I wanted to highlight the upcoming webinars that we have from EMBL-EBI. So next week we have navigating patent data with Shaw Kemble, and following on the week after we have calculating polygenic scores with the polygenic score catalog calculator. So those are both available to register for now. Right, okay, so I think we can probably go straight on to the questions at this point, as we have a few ready waiting for us. Okay, so we have a question that came up when I think Francisco, you were taking us through the site. What does manually marked as significant mean? So what are the criteria used to manually mark those phenotypes where the p-value is unavailable?
So I guess that will depend on each parameter. So the manually marks as significant phenotypes, those are the ones that come directly from the mouse clinics. This is because this type of data is not processed by the statistical pipeline, so it doesn't produce a p-value. So to know about this criteria, you will need to go directly to the IMPRES description to view exactly about the details and how it's measured or considered for each procedure.
Thank you. We also have a question that came up when Diego, you were looking at the predicted disease associations, and I think in fact this came in a bit before you were talking in more detail about Phenotyping, but they were saying it wasn't quite clear about where the predicted disease associations came from in that second tab on the human disease section. Could you maybe show in a little more detail?
Of course. So when we have associations or when we have a specific disease in a specific mouse model for a specific gene, there might be a case where the disease has not been associated to that specific gene. But there are mouse models that might have phenotypes that resemble those phenotypes seen in the human disease. So that's what we call the predicted associations, when the phenotypes of a specific disease match the phenotypes of a mouse model, even if the gene corresponding to that mouse model is not directly associated with that disease in any the curation and entities that we use to build the pipeline.
Okay, great. And we have another question that's about choosing strains. So how many strains do you use for studying the phenotype of each gene? I think maybe sort of flip the wrong way around. How do you choose which strains to use for which genes?
So the IMPC works on a standard genetic background. So all of the mutant strains are made on a C57 Black 6N background. The production is aimed at producing a knockout of the gene function. Quite often the aim is to actually remove a sort of critical exon or a set of critical exons that remove gene function completely. And so those are the alleles that are actually analyzed. And historically, IMPC started analyzing alleles produced by ES cell based techniques, and that switched over about 5 or more years ago to CRISPR production of the alleles. So we have a variety of different types of alleles on the website.
Okay, great, thank you. Sorry, just reading one that came in. So, Francisco, you mentioned allele names that include things like 1A and 1B in them. Is that something you're able to elaborate a bit more on?
So both of those alleles are ES cell based alleles, and the TM1B allele will be derived from a TM1A allele, which involves excising some marker genes. And there's a page on the website that explains all of the different types of alleles that are studied by IMPC. You'll find that in the top menu under About IMPC. I think it's a section called Allele Design.
Right, okay. I think we have one more question around Phenotyping. So the question is, how does Phenotyping weigh different matching and non-matching phenotypes?
So that depends on how closer the HP term, so the term in the disease, and the MP term in the mouse are in the ontology. It basically depends on that.
Okay, great, thank you. Right, so I think in that case, if we don't have any more coming in, sorry, one more question before we let you all. Lots of questions coming in now. So you have your p-value set to 10 to the -4. Is there a particular reason you've chosen to do it that way rather than let sort of users affect their own p-value?
If you'd like to use another threshold, for example, 10 to the -2, you can access our data programmatically and change a filter. 10 to the -4 filter is used because it's powerful threshold enough, and yeah. I think the better way to set other p-value is to by downloading the data and applying filter.
That's great, thank you. And as was mentioned before, Marina's written and created an online tutorial all about using programmatic access to access the IMPC data. My link to that is in the chat if that's something anyone would like to learn more about. There's also a question again about Phenotyping, about weaknesses or limitations of it. Is that something you're able to tell us a bit more about, Diego?
Sure. So I think potentially the greatest weakness is because the score goes down when the mouse is not able to capture those disease phenotypes. If the disease has loads and loads and loads of phenotypes, for every phenotype that's not captured in the mouse, that score might go down. So you might end up with perhaps visually low scores, but it doesn't really mean that it's a low score. It's just because the disease has loads of phenotypes. And the second drawback is diseases keep getting more and more HP annotations through time, and perhaps more rapidly than mouse models. So there could be a mismatch between perhaps mouse models will not be able to capture those same or the equivalent HP terms from the disease as quickly as we would like to, so that we can present the score in a timely manner.